Adenylate kinase. Adenylate kinase EC2.7. 4.3, also known as ADK or myokinase, is a phosphotransferase enzyme that catalyzes the interconversion of the various adenosine phosphates ATP, AD, and AP. By constantly monitoring phosphate nucleotide levels inside the cell, ADK plays an important role in cellular energy homeostasis. Substrate and products. The reaction catalyzed is AP plus AMP equals 2AD. The equilibrium constant varies with condition, but it is close to 1. Thus, DGO for this reaction is close to 0. In muscle from a variety of species of vertebrates and invertebrates, the concentration of ATP is typically 7-10 times that of ADP and usually greater than 100 times that of AMP. The rate of oxidative phosphorylation is controlled by the availability of ADP. Thus, the mitochondrion attempts to keep ATP levels high due to the combined action of adenylate kinase and the controls on oxidative phosphorylation. Isozymes. To date, there have been 9 human ADK protein isoforms identified. While some of these are ubiquitous throughout the body, some are localized into specific tissues. For example, ADK7 and ADK are both only found in the cytosol of cells, and ADK7 is found in skeletal muscle, whereas ADK is not. Not only do the locations of the various isoforms within the cell vary, but the binding of substrate to the enzyme and kinetics of the phosphoryl transfer are different as well. ADCone, the most abundant cytosolic ADK isozyme, has a Km about 1,000 times higher than the Km of ADK7 and 8, indicating a much weaker binding of ADCone to AMP. Subcellular localization of the ADK enzymes is done by including a targeting sequence in the protein. Each isoform also has different preference for NTP. Some will only use ATP, whereas others will accept GTP, UT, and CTP as the phosphoryl carrier. Some of these isoforms prefer other NTP entirely. There is a mitochondrial GTP, AMP phosphotransferase, also specific for the phosphorylation of AMP, that can only use GTP or ITP as the phosphoryl donor. ADK has also been identified in different bacterial species and in yeast. 2 further enzymes are known to be related to the ADK family, i.e. yeast uridine monophosphokinase and slime mold UMP-CMP kinase. Some residues are conserved across these isoforms, indicating how essential they are for catalysis. One of the most conserved areas includes an ARG residue, whose modification inactivates the enzyme, together with an ASC that resides in the catalytic cleft of the enzyme and participates in a salt bridge. Subfamilies. Adenylate kinase, subfamily interpro, IPR 6259, UMP-CM, B kinase interpro, IPR 6266, adenylate kinase, isozyme 1 interpro, IPR 6267. Mechanism. Phosphoryl transfer only occurs on closing of the open lid. This causes an exclusion of water molecules that brings the substrates in proximity to each other, lowering the energy barrier for the nucleophilic attack by the phosphoryl of AMP on the G-phosphoryl group of ATP. In the crystal structure of the ADK enzyme from E. coli with inhibitor of PHIV-A, the RJT8 residue binds the PHIV-A at the phosphate group. It has been shown that the mutation RIT8G results in 99% loss of catalytic activity of this enzyme, suggesting that this residue is intimately involved in the phosphoryl transfer. Another highly conserved residue is argon 119, which lies in the adenosine binding region of the ADK and acts. It has been suggested that the promiscuity of these enzymes in accepting other NTP is due to this relatively inconsequential interactions of the base in the ATP binding pocket. A network of positive, conserved residues Lis13, argon 123, argon 156, and argon 167 in ADK from E. coli stabilize the buildup of negative charge on phosphoryl group during the transfer. 2 distal aspartate residues bind to the arginine network, causing the enzyme to fold and reduces its flexibility. A magnesium cofactor is also required, essential for increasing the electrophilicity of the phosphate on AMP, though this magnesium ion is only held in the active pocket by electrostatic interactions and dissociates easily. Structure, flexibility, and plasticity allow proteins to bind to ligands, form oligomers, aggregate, and perform mechanical work. Large conformational changes in proteins play an important role in cellular signaling. Adenylate kinase is a signal transducing protein. Thus, the balance between conformations regulates protein activity. ADK has a locally unfolded state that becomes depopulated upon binding. A 2007 study by Whitford et al. Shows the conformations of ADK when binding with ATP or AMP. The study shows that there are three relevant conformations or structures of ADK core, open and closed. In AD, there are two small domains called the LID and NMP. ATP binds in the pocket formed by the LID and core domains. AMP binds in the pocket formed by the NMP and core domains. The Whitford study also reported findings that show that localized regions of a protein unfold during conformational transitions. This mechanism reduces the strain and enhances catalytic efficiency. Local unfolding is the result of competing strain energies in the protein. The local thermodynamic stability of the substrate binding domains at PLID and AIM-PLID has been shown to be significantly lower when compared with the core domain in ADK. Coli. Furthermore, it has been shown that the two subdomains at PLID and AIM-PLID can fold and unfold in a non-cooperative manner. Binding of the substrates causes preference for closed conformations amongst those that are sampled by ADK. These closed conformations are hypothesized to help with removal of water from the active site to avoid wasteful hydrolysis of ATP, in addition to helping optimize alignment of substrates for phosphoryl transfer. Furthermore, it has been shown that the enzyme will still sample the closed conformations of the ATPLID and AIM-PLID domains in the absence of substrates. When comparing the rate of opening of the enzyme, which allows for product release, and the rate of closing that accompanies substrate binding, closing was found to be the slower process. Function, metabolic monitoring. The ability for a cell to dynamically measure energetic levels provides it with a method to monitor metabolic processes. By continually monitoring and altering the levels of ATP and the other adenyl phosphates ADP and AMP levels, adenylate kinase is an important regulator of energy expenditure at the cellular level. As energy levels change under different metabolic stresses, adenylate kinase is then able to generate AMP, which itself acts as a signaling molecule in further signaling cascades. This generated AMP can, for example, stimulate various AMP-dependent receptors, such as those involved in glycolytic pathways, KATP channels, and 5-AMP-activated protein kinase AMP. Common factors that influence adenine nucleotide levels, and therefore ADK activity, are exercise, stress, changes in hormone levels, and diet. It facilitates decoding of cellular information by catalyzing nucleotide exchange in the intimate sensing zone of metabolic sensors. ADK shuttle. Adenylate kinase is present in mitochondrial and myofibrillar compartments in the cell, and it makes two high-energy phosphoryls B and G of ATP available to be transferred between adenine nucleotide molecules. In essence, adenylate kinase shuttles ATP to sites of high energy consumption and removes the AMP generated over the course of those reactions. These sequential phosphotransfer relays ultimately result in propagation of the phosphoryl groups along collections of ADK molecules. This process can be thought of as a bucket brigade of ADK molecules that results in changes in local intracellular metabolic flux without apparent global changes in metabolite concentrations. This process is extremely important for overall homeostasis of the cell. Disease relevance. Nucleoside diphosphate kinase deficiency Nucleoside diphosphate in DP kinase catalyzes in vivo ATP-dependent synthesis of ribo and deoxyribonucleoside triphosphates. In mutated Escherichia coli that had a disrupted nucleoside diphosphate kinase, adenylate kinase performed dual enzymatic functions. ADK complements nucleoside diphosphate kinase deficiency. Hemolytic anemia, 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 anemia. Adenylate kinase deficiency in the erythrocyte is associated with hemolytic anemia. This is a rare hereditary erythromyopathy that, in some cases, is associated with mental retardation and psychomotor impairment. At least 2 patients have exhibited neonatal icterus and splenomegaly and required blood transfusions due to this deficiency. In another patient, an abnormal fragment with homozygous and heterozygous G substitutions at codon 164 caused severe erythrocyte ADK deficiency. 2 siblings had erythrocyte ADK deficiency, but one did not have evidence of hemolysis. A cone and post-ischemic coronary reflow. Knockout of a cone disrupts the synchrony between inorganic phosphate and turnover at ATP-consuming sites and ATP synthesis sites. This reduces the energetic signal communication in the post-ischemic heart and precipitates inadequate coronary reflow flowing ischemia reperfusion. ADK2 deficiency. Adenylate kinase 2A2 deficiency in humans causes hematopoietic defects associated with sensorineural deafness. Reticular dysgenesis is an autosomal recessive form of human combined immunodeficiency. It is also characterized by an impaired lymphoid maturation and early differentiation arrest in the myeloid lineage. A K2 deficiency results in absent or a large decrease in the expression of proteins. A K2 is specifically expressed in the stria vascularis of the inner ear, which indicates why individuals with an K2 deficiency will have sensorineural deafness. Structural adaptations. A cone genetic ablation decreases tolerance to metabolic stress. A cone deficiency induces fiber-type specific variation in groups of transcripts in glycolysis and mitochondrial metabolism. This supports muscle energy metabolism. Plastidial ADK deficiency in Arabidopsis thaliana, aliana. Enhanced growth and elevated photosynthetic amino acid is associated with plastidial adenylate kinase deficiency in Arabidopsis thaliana.